Hi everyone, welcome to my YouTube channel Biology Tips. In today's session, we are going to discuss about exons and introns. It's an important topic in molecular biology. So, before going in depth about exons and introns, you should know what you mean by split genes. So, what are split genes? Eukaryotic genes are called as split genes, as coding sequences called exons are interrupted by the non-coding sequences called introns. So, most of you know about this concept. Split genes are present in eukaryotes because they possess coding sequences and non-coding sequences in their gene. Coding sequences are called as exons and non-coding sequences are called introns. So look at this diagram. Here you can see this is one exon, followed by that there is an intron, then there is an exon, right? Moving on to the next session, we are going to study what are introns. So intron is a word coined by Walter Gilbert in 1978. He coined the term intron refers to part of primary transcript that is not included in a finished mRNA, rRNA or tRNA. So what are introns? Introns are the non-coding sequences. It will be present in the DNA, but when you make hnRNA, the primary transcript, right, it will have this introns. But when you make a mature RNA, mature mRNA, then what will not be there? Introns won't be there, right? So that's why he's telling introns refers to part of primary transcript that is not included in a finished mRNA, rRNA or tRNA. Now, introns are the non-coding sequences. It will not appear in the mature or processed RNA. It will not code for proteins. Introns are removed by recognition of the donor site at the 5' end and the splice acceptor site at the 3' end. So, what are introns? Introns are the non-coding sequences. The term is coined by Walter Gilbert. He coined this term in 1978. Now, what he told, introns are part of DNA or part of the hnRNA, which will not be present in the finished mRNA, tRNA or rRNA. Moving on to the next session, we are going to study about exons. So what are exons? Exons are the coding sequences which is present in the mature RNA, right? So exons are the coding sequences or express sequences that appear in mature or processed RNA. Exons provide instructions for coding proteins which create mRNA necessary for the synthesis of proteins. So these type of questions normally in +2 level it can come for 1 mark, right? When you look at the higher level, right? This is part of molecular biology. This is part of the essay question transcription. Moving on to the next session, we are going to see what do you mean by splicing. Splicing is a process by which we are making the mature RNA, because the primary transcript which is formed after transcription, it is having introns. So we are calling it as the hnRNA or the heterogeneous nuclear RNA, right? So hnRNA possesses exons and introns. Now we have to remove these introns. These introns are removed by a process called splicing. Now, who helps in splicing? Spliceosome. The primary transcript contains both exons and introns. Hence, it is subjected to a process called splicing, where introns are removed and exons are joined in definite order to form mRNA. Definite order to form mRNA. The removal of introns are promoted by small nuclear ribonucleoprotein particles, snRNPs, pronounced as SNRPs. SNRPs are formed by the association of small nuclear RNA with proteins. Spliceosome represent the snRNP association with hnRNA at the exon-intron junction. Okay. So, SNRPs are formed by the association of small nuclear RNA with proteins. Right? Now, spliceosome represent the snRNP association with hnRNA at the exon-intron junction. So, look at this process. Look at this picture. This is a method of splicing which occurs inside the nucleus. Correct? Because split genes are present in eukaryote. When you look at eukaryote, transcription occurs inside the nucleus. Now, after transcription, what you are getting? It is called as the hnRNA or the heterogeneous nuclear RNA. So the splicing has to occur inside the nucleus. Now, who helps in splicing? snRNPs. We will call it as SNRPs, the small nuclear ribonucleoprotein particles, which are formed by the association of small nuclear RNA with protein, right? Small nuclear ribonucleoprotein particles. From the name itself, you know, they possess small nuclear RNA with proteins, snRN plus proteins. They possess small nuclear RNA plus proteins, right? Now what the SNRPs will do? They will come and join with the junction of exon and introns here. Once again here, right? They will join at the junction of exons and introns. So here it will join, right? Now what will happen? Once it is joined, right? It is called as the spliceosome. Now this intron part is looped out and it is removed, right? Here you can see the intron part is looped with the help of spliceosome. Now what is spliceosome? The joining of snRNPs with introns, right? Once it is looped out, it is excised. Now this is the excised intron. The remaining exons, they are joined again. Now what you are getting? This is the mRNA, right? Okay. So this is a process by which we are removing what? We are removing the introns. The primary transcript contains both exons and introns. Hence, it is subjected to a process called splicing, where introns are removed and exons are joined in definite order to form mRNA. The removal of introns are promoted by small nuclear ribonucleoprotein particles, pronounced as SNRPs. SNRPs are formed by the association of small nuclear RNA with proteins. Spliceosome represent the snRNP association with hnRNA at the exon-intron junction. This process occurs in the nucleus. Now, till now we studied about the normal mechanism of transcription. Normal mechanism of transcription means from one hnRNA we are producing one mRNA, but there is complex transcription. From one hnRNA we can produce more than one protein. The term introns and exons are meaningful when they are applied to a primary transcript that give rise to only one mRNA molecule. Complex transcription units have multiple splicing arrangements. So in the coming session, we will study about the complex transcription. In complex transcription, there is a method we use called alternate splicing. Okay. Now, what is alternate splicing? Alternative splicing or alternative RNA splicing or differential splicing is a regulated process during gene expression that results in a single gene coding for multiple proteins. This process results in different mRNA molecules which can produce different proteins. So, look at this picture. Here you can see this is a gene which contains exon 1, exon 2, exon 3 and exon 4. Here you can see introns are present, right? Now, once this hnRNA is formed, that time also you can see that, right? What is there? We have the introns. Now, in alternate splicing, you can see that exon 1, 2 join with 3, whereas there is protein B is formed by taking exon 1, 2 with exon 4, right? So, we are producing two proteins, protein A and protein B from the same gene, right? So, this method we will call it as the alternate splicing. This process results in different mRNA molecules which can produce different proteins. The proteins translated from alternatively spliced mRNAs will contain differences in their amino acid sequence and their biological functions. This process results in mRNA heterogeneity. It helps in the regulation of gene expression. How it helps in gene regulation? Sometimes we require only protein A, so only protein A will be formed. Sometimes we require protein B, so protein B will be formed, right? Accordingly, we will be synthesizing the mRNA. So when we require the mRNA for protein A, that time the mRNA for protein A will be formed. When we require mRNA for protein B, that time the mRNA for protein B will be formed. Okay? Moving on to the next session, we are going to study about faulty splicing. Splicing of DNA has to be performed with precision to produce functional mRNA. Faulty splicing, as the name suggests. Somewhere the splicing went wrong. So splicing of DNA has to be performed with precision to produce functional mRNA. Faulty splicing results in disease. Example, beta thalassemia. A method where the beta polypeptide in the case of hemoglobin is not formed, right? So in today's session we studied about exons, introns, split genes, splicing, spliceosome and faulty splicing. Okay, so I hope you are able to follow. Thank you. Please subscribe my channel